I announced Umberto Simeoni. I'm very, very happy that he came here. He's one of very, very kind neonatologists. All neonatologists are kind in general. He's especially kind. And we met in Paris when we had to audit the Necker Hospital in Ville for some certification or so. But of course I knew his literature, I admired all his work, and I'm really grateful that he came to give a lecture, and you will see what he has to tell us. Thank you for coming.
Thank you so much. Really, it's my pleasure, and so I find this meeting extremely inspiring. The interdisciplinary discussions. So I guess we are moving back to the topic of the first session this morning about perinatal and neonatal diseases. Are they a particular risk for later disease and in the offspring, mainly cardiovascular or metabolic? We know today that preterm-born infants, when they become adults, for example, have increased arterial blood pressure. This is demonstrated by this meta-analysis, but a number of studies have confirmed this finding, including by the group of Neil and Modi. And they have a slightly higher blood pressure, especially women. But we know today that such a small difference at the age of 20 or 30, 3-4 millimeters mercury, means a lot for later health of these subjects, because such a difference can increase the risk of later stroke by 30%. So there is no difference depending on the cause of preterm birth, except maybe that preeclampsia shows a bit higher effect. I don't know if here, a bit higher blood pressure compared to other causes, but it seems independent on the context, on the cause of preterm birth. Not only we have also a long list, which is increasing, of long-term disorders at adulthood, which have been associated with preterm birth in more or less all the systems or the organs, including metabolic disorders with altered glucose tolerance, altered renal function in some patients, respiratory disorders, of course, also behavior, personality, mental health are affected, also reproduction, and the lifestyle also are influenced by preterm birth. Surprisingly, if we look at other major obstetrical syndromes, like intrauterine growth restriction, or to the contrary, macrosomia due to maternal diabetes, for example, we find very similar outcomes at adulthood, with an increased risk of hypertension and of metabolic diseases at least. And these are the key points for later health, of course, of these subjects. And not only the consequences are similar, but the mechanisms are remarkably similar, despite different starting points. Because, of course, there is no comparison possible in terms of nutrition between a macrosomic infant and a low birth weight, an extremely low birth weight infant due to preterm birth, complicated by fetal growth restriction. The mechanisms, we know them at least grossly at the physiologic level or even at the cellular level. And for hypertension, there are both renal changes, which are characterized with a reduction in the nephron number, which we have for life. We acquired the number of nephrons in the last part of pregnancy, and then we keep them for life. And of course, we know today that the number of nephrons, the nephrons endowment, correlates with the birth rate in particular. So of course, with less nephrons, each nephron has to work more, and glomerular sclerosis develops early. There are vascular causes as well, especially endothelial dysfunction, both in angiogenesis, the vasodilating capacity, especially endothelium-dependent vasodilating capacity, arterial stiffness, intima media thickness. There is also a reactivity to stress, which is altered. On the metabolic side, both reduced sensitivity to insulin due to changes in body composition, likely, and insufficient insulin production can increase this risk. But these mechanisms are found in all these perinatal situations. So the question we may ask, I come closer, it moves, is that if we look now not as at the patients who suffered perinatal complications, but at the general population, we have a pandemic evolution of what we call non-communicable diseases of adults, such as hypertension again, but here is the evolution of the number of adults with diabetes between 1980 and 2014, which with a dramatic increase, and with the colors which are close to the red and yellow are the regions in the world which have the highest numbers. And these regions are today mainly low-income countries in Africa and Southeastern Asia. So this spread, this evolution is not due to, it's not a genetic problem, clearly, it's environment. And the question may be, and we studied the long-term consequences of, for example, of preterm birth or the obstetrical complications, to check whether this can be kind of a model of what happens, an extreme model, unfortunately, of what happens in the general populations. Because today we know that these diseases have a number of origins, but an important part is acquired very early in life. And it is possible that perinatal disorders are part of an extreme part of this pathway, early origins of disease. Nevertheless, they may act as well as a stress test for previously acquired conditions in the mother or in the father, and reveal some antecedents of cardiovascular risk simply due to the stress of pregnancy, especially in the case of complications. This I don't know, but I'm convinced that they enter in this picture, global pictures of the importance of the interactions between the early environment and the genome in the development of the child, with long-term consequences. Of course, they have sometimes also part of causal factors which are simply, unfortunately, damage and lesions, but most of them are functional. So to understand better this, we can follow the classical scheme which was developed by Waddington. He called this epigenesis, and he understood very early, before we knew so much as today about the sequence of the genome or epigenome, that the trajectory of development and this sphere represents a stem cell, but it may also represent an individual. And it's simply, the important point is that either the cell or the individual can follow several trajectories in life, which are represented by the slope which this bowl chooses to come down. And the trajectory depends on the mountains and the valleys, which are represented here, and which represent the environment. So one genome can result in an infinity of phenotypes. Also, the genome is important. Environment adds an enormous effect in terms of diversity. Now when I press one, it's like so. Sorry. So, this is called a named developmental programming, which is an excessive term because it's not a determinism, it's an influence, a risk factor, or a positive factor for health, which is acquired very early. Two examples to illustrate it. On the left, a very simple experiment in rat pups. This is their growth in weight with an extremely simple intervention, which is reducing their food for 3 weeks, but at two different times during growth and development. On the right, between 9 and 12 weeks. I don't know if you can see it in the back, but these animals, when the nutrition is reduced, of course, they alter their growth curve, and when food becomes again normal after 3 weeks, they catch up in terms of growth and gain growth to reach their corridor. But if the intervention is performed earlier, during early development, between 3 and 6 weeks, their growth velocity slows down. They start catching up, but they reach a different set point, which is a different growth corridor. So this is why stimuli in this particular period of the beginning of development, we call it in the human the 1,000 days, between conception and the second anniversary of the baby. And so the difference between the two experiments is the timing only. The intervention is same, and this shows the sensitivity of the early development to the environment, here nutrition. And the second observation is the classical work by David Barker and his group. Here is for the risk for death due to coronary heart disease, which correlates, of course, with birth weight. The lowest the birth weight, the highest the risk. We know today that this curve is indeed a U-curve, and macrosomia leads to the same consequences. But what is important is to note that these birth weights were mostly within the normal range. So even slight variations in the environment at this time period, can have long-term important consequences. We understand this as the fact that the infant or the fetus, while it is developing, develops not by simply reading the genetic code, but by interpreting the signals it receives from the environment and virtually trying to adapt its development through developmental plasticity to the predicted environment in which he will or she will live for the entire life. So if these signals are not optimal, if they are biased, the fetus may set up its regulations at a suboptimal level for arterial blood pressure or blood glucose control, for example, any physiologic function. So if there is an adverse environment during the period of vulnerability, the window of sensitivity of early development, the setting of the regulation may not be optimal, especially if later the environment becomes more favorable or even excessively affluent. The regulations at this age are set as the definition of programming for the long term, and the stimulus has a long-term effect and not only a short-term one. So the mechanisms, as far as we know, in the general populations are same as those I mentioned for preterm birth or macrosomic infants about hypertension or glucose tolerance at the beginning of this talk. And we can understand the story as here is the life course, the period of sensitivity during early development, and adult life with a latency period between both. And among the circumstances which are able to alter the normal programming of physiologic functions which occurs early in life, preterm birth, IUGR, etc., all perturbations of the perinatal period in particular can have long-term effects likely as a kind of exaggeration of the normal variations in the environment. And this might be an explanation of what we observe today in these patients. But all the other circumstances can alter programming, including what happens in the pre- and periconceptional period. Mireille stressed this morning that intervention which occur too late in pregnancy are unsuccessful in terms of prevention, acting on the lifestyle. And so the very early times, including before conception, are extremely important. And this is due to, we think today that it's likely due to epigenetic changes among other factors. Epigenetics simply are a way to modulate gene expression without changing the gene sequence. So, according to the stimuli received by the environment, epigenetics translates them into changes in the environment of the DNA. And these changes at the molecular level or at the level of the chromatin just set the regulation of the activity of a specific gene up or down, but on the long term, for decades. It's not like a transcription factor which stimulates the gene in a timing of hours. This is for decades, eventually lifelong, and can even be inherited by the next generation, although it is an acquired trait, which was quite surprising because we knew that only genetic changes can be inherited, while acquired traits through the inheritance the transgenerational inheritance of epigenetic marks can be transmitted through 2, 3, maybe 4 generations. These changes are mainly methylation of DNA, changes in histone conformation, and non-coding RNAs. So they affect the regulation of genes, the synthesis of proteins, and they may explain the lasting effects of developmental plasticity. They are flexible, dynamic, reversible, and stay over the life course. And the development itself is epigenetic in nature, because the stem cell of Waddington, to commit itself to become a neuron or an epithelial cell, simply needs to silence part of its genome, which is not needed for this fate, and enhance the part of the genome which will be used in neurologic function or epithelial function, as every cell in the body has the whole genome. And epigenetics so are key in development, and that might be why this period of early life is so sensitive to the early development. We can therefore understand this phenomenon as very similar to what you showed this morning, Mireille. Here is the life course, and the risk of alteration for physical, mental, or social health or well-being. We have here the period of 1,000 days and 3 trajectories which are related to 3 different environments. 1 healthy in green, and 2 unhealthy in blue and red. And of course, what happens in the first 1,000 days induces important changes later in life. The differences are very small here, but they are larger later, and other stimuli occur, of course, in life. And based on the same general trajectory, to make it simple, we may imagine that the exposure to subsequent adversities just changes the position of the individual with respect to the general trajectory. And this is due to the epigenome. The effect of the genome, especially about non-communicable diseases, is existing. It's a real effect, but we know today that it's about 5 or 10% of these cases which are explained by a particular gene in obesity, diabetes, or hypertension, for example. But this allows us to have a new definition of predisposition, which is not only the genome, as we thought a few decades ago, but it includes the epigenome, which is certainly as well important. And this, in terms of prevention, has important effects, as it is relatively easy to change the trajectory early in life, or simply to avoid an unfavorable trajectory by early prevention. It is much more difficult to change this, to change our behaviors, or the behavior of our patients later in life, especially at an age which is more mature, when the lifestyles are very difficult to change. So developmental plasticity is maximum at the beginning of life, of course, and decreases, while the inability to respond to health challenges increases. And one final important point is this transgenerational possible effect. We have here a representation of what happens with the first generation and the second generation. The early environment, including stress, nutrition, the exposure to toxicants, and lifestyle, including the lifestyle of the parents, of course, induces long-term favorable or unfavorable consequences to the offspring, thanks to a basic mechanism which is likely to be epigenetic, which affects behavior, emotions, vulnerability, social vulnerability, metabolic, hypertension, cardiovascular health, and neuropsychic diseases, among others. But due to the possible inheritance of acquired epigenetic marks, the next generation is affected as well. And what is important is that this can be a leveraging effect, which could explain part of this terrific increase in the number of diabetic persons, for example, worldwide. And these diseases kill dozens of million people each year in the world and have a tremendous cost, of course. So it's important to act early, and just to keep in mind that the size of this effect is not simply anecdotal. It's not a scientific curiosity, or it's really important because in the general population here in Finland. Here is the frequency of hypertension according to the current weight at adulthood. Of course, the frequency increases with an increased weight at adulthood, but the effect of birth weight here is extremely important and comparable to that of the weight at adulthood, simply between more than 4 kilos and less than 3 kilos. At birth. It's a very similar influence. If we take into account that the weight at adulthood can be also partly programmed very early in life, we know that obesity has also the same early origins, it is certainly worthwhile.
Sorry, I don't understand that, because 3 kilograms of birth weight,
yeah.
The prevalence of hypertension is higher.
Yes, because when the birth weight decreases, the risk for cardiovascular and altered cardiovascular function.
Is 3
yes,
3 kilograms of
well, here, no, it's kilo. And the limit is here. Here we are below 3 kilos, but it's true, it's the results of this.
That means that big babies more than 4 kilos.
And above, yes, this study doesn't show this, but it is likely that it will increase as well. Other studies have shown this. Yeah.
But we know that if you're born big and you're not big later in life, that's the ideal setting.
Yeah.
Other researchers have shown that.
Yes, and of course, it's a statistical risk. It's not a determination of disease. What happens later is important as well, and especially the lifestyle. So I would like to stress simply also that, yes, just stress is extremely important. You may not be able to read this, but it's a list of the consequences of exposure to stress early in life and to adversity, especially during infancy. I will read it. All these differences, nearly all are significant, statistically significant. They include physical inactivity later, the risk of having low physical activity, overweight or obesity, diabetes, cardiovascular disease, heavy alcohol use, cancer, some cancers, liver diseases, becoming a smoker. respiratory disease, multiple sexual partners, anxiety, teenage pregnancies, depression, etc. And the highest violence victimization and violence perpetration, which are kind of the part of the, of course, of the social well-being. And the odds ratio are around 7 or 8 in this case, so it's not a light effect. So this simply means that both in the general population, but also maybe in patients who suffer perinatal disorders, we must be aware of that, and public health policies should take this into account and offer support, follow-up, some information to these patients and their families, and special attention. Finally, I would like to simply to show you that there are possibly some common mechanisms, even between, not only between organs and systems, but also at the molecular level. And in our approach, we found in numerous models, in human and in various species, that stress-induced accelerated cell senescence is a common feature of these circumstances of altered early programming, starting here in the endothelium from preterm infants, studying endothelial progenitor cells circulating in the cord blood of preterm infants. We found that they have a reduced capacity to proliferate, to build new vessels, as shown here in this experiment of in vivo transplantation of stem cells from human preterm infants on the left, and stems and endothelial progenitor cells from controlled healthy newborn on the right into nude mice. And you can see that the endothelial progenitors from healthy newborn are able to form a nice vessel, while those of the preterm infants are not. This may result in a reduced vascular bed, which may be long-lasting and possibly be an explanation of increased arterial resistance and risk for hypertension. There are changes in pro-angiogenic, angiogenic factors, but what is interesting is that in these endothelial progenitor cells, there is an accelerated senescence, which is related to a deficit in sirtuin-1, which is a key factor in controlling cellular senescence, and this deficit is related to a change in the histones, which are related to sirtuin-1, which is an epigenetic change in these cells in preterm infants. Interestingly, the group of Ingram in the US has found similar results. This is the capacity to build vessels in vitro, in matrigel, in endothelial progenitor cells from the cord blood of infants exposed to maternal diabetes, the opposite condition. Again, very similar mechanisms. And simply one example in one of our models, animal models in mice, by simply overfeeding mice pups during the period of lactation, by reducing the number of pups in the litter with an increased access to breast milk from the mother, these mice develop hypertension at adulthood, altered renal function, and kind of fatty liver disease. And this is associated as well with an accelerated senescence, cellular senescence, as shown by these various markers, including again with sirtuin-1 which is altered. So we have the feeling that it is paradoxically important. It is important to consider that paradoxically these circumstances very early in life lead to an accelerated senescence, even including in preterm infants. And there is a correlation with stress because we studied this, for example, in the preterm infants in cold blood, so there was no nutritional intervention. The only factor could be stress of being born preterm. In a completely different field, the group of Yehuda, you may know, have studied the consequences of stress due to having been exposed to the Holocaust and being born to an Holocaust survivor during the last World War. And these persons have some disorders of depression, anxiety, which are often associated with altered glucose tolerance. And this, again, is a mechanism which is not surprising, as the stress is mediated by the hypothalamic pituitary adrenal axis and cortisol, which both regulate stress reactions and as well glucose metabolism. And the gene of the receptor to glucocorticoids in these subjects has a different methylation compared to control. So these also are likely to be epigenetic marks. I finish with this last example of the importance of stress early in life with consequences. This is again in animals, performed in Zurich by the group of Gap and Monsui. And they stressed mice mothers during gestation, and then they continued stressing mothers and pups by unpredictable separation, temporary separation of the pups from the mother. And they observed that, especially in the male offspring, the next generation had different behavior and mental health, with depression again, altered behavior in at-risk situation associated with glucose metabolism disorders, and especially poor glucose tolerance. And this was associated with a particular profile of micro non-coding RNAs in the sperm of this generation. And microRNA, non-coding RNAs are, aside the methylation of DNA and the changes in the histones, the third factor which is involved in epigenetic change. They regulate, of course, the activity of genes. It's a major part of our RNA. And using a specific microRNA from the sperm of these F1 generation males, injecting it into a normal ovocyte fertilized by a normal spermatozoid from a normal mice, they were able to reproduce the phenotype of this newly conceived individual. So it's important, and the father has also an extremely important influence, especially preconceptionally. So to end, what is interesting in this concept or in this approach is of course the possibility of prevention. Knowing this, it is much, I would say, much more interesting to act early with preventative interventions on the environment, on lifestyle, especially if we come in early gestation, including from health economics point of view, as shown here by the studies by Heckman, the return to investment in human capital increases exponentially when you act early in life. And it's no surprise. This has been stressed in a Lancet Papers series published last year. And here Tom Fleming nicely showed the various stages of, and the importance of both paternal and maternal factors conditioning the development in the periconceptional period, and including in vitro fertilization. It is the changes induced are likely to be more important in the preconception periods than later, even in this 1,000 days period. And finally, two examples which are just happening in France, where I was working a few years ago. And we were invited just to give a talk on this by a health insurance company. And then they were interested in the concept, and we met a few times, and by themselves they decided to apply this, where we would have considered that knowledge is insufficient to define the best strategy, the best intervention, how to evaluate it. They after a few months, they told us that they have started offering to the person they were the insurers, and it was a branch of their affiliates, which was the people who were working to make bread, pastries, to sell them, which are considered at risk of overweight and obesity. And they offered this spontaneously to these people in the package of health insurance, of complementary health insurance. In French, it is still working. This includes a preconception intervention, which is a sensitization information, with a preconceptional consultation, and then a follow-up online, a follow-up by a particular team which has been trained specifically over pregnancy and postnatally to promote breastfeeding based on a personalized approach. And they put the money, and they told us that for them it's clear that they spend less by this prevention, early prevention, than, of course, curing the consequences of unfavorable lifestyles later. And also, it's of note, the French government has started a program based on the 1,000 days, which is supported currently by a specific commission, principally oriented toward psychosocial health, and with a series of measures. We are waiting for what they will be, and working on this as well, including to focus on parents and to allow them, to empower them to be able to adapt their lifestyle, and especially promoting breastfeeding and rendering it possible. And this is still a work we just started in October, and it's interesting anyway. I guess it's a positive finding. It will be certainly difficult to evaluate in the effects, but at least it is possible to act, and not only to, of course, to make recommendations. And sometimes doing it, just do it, can be a way to make some progress. That's what I wanted to say.
Any questions?
So I was really nice talk. Thank you very much. So I have several questions. So first question is, can we expect in the future, when we see the trends in assisted reproduction, that we should deal with a new group of potential problems, healthy problems in the population?
Well, not a new one, but it's a risk factor among others, as far as we know today, because we know that these infants born after IVF, for example, are at increased risk. The same story later of hypertension, etc. A number of disorders. It's I would say a light increase, as far as we know today. But of course, if we look at the long term, it may be significant at the level of a population. It may have important consequences. It's very difficult to understand if it is due to the lifestyle or the environment of the parents before they wanted to have a baby, because there was an infertility, and the cause could be environmental of this infertility. So is it the cause of the problem which leads to these long-term consequences, or the intervention of ART? And then also the consequences are also biased by the fact that there are still some multiple pregnancies which of course affect intrauterine nutrition, sometimes induce preterm birth, and this is also bias in the observational studies at least. So we know little at the moment, but I would say that we know little in the whole field, and it's a rather complex issue, but some observation in the
Well done.
Do you have another question?
I have one question more.
Yeah.
So we can find some articles, and they explain the incidence, the high incidence of diabetes in the population and obesity. They try to explain it with the increasing incidence of cesarean section and with the microbiome. What do you think? Have we fortunately enough data that we confirm it?
Well, the whole story is difficult to confirm because it's based on observation studies mainly, and we have some interventions which are being performed, including randomized controlled trials. But of course, it's observational. I don't know if propensity score could help, certainly. But then it's based on epidemiology first. So I take the example of sudden infant deaths. We reduced it by 60%, the incidence, not based on randomized trials or simply changing the position of the baby during sleep, and on a pure observational approach, and it worked very well. Same for the maximum speed on the roads. Not everything is a matter of randomized controlled trials, but for C-section it's difficult to say that it changes the life of. There are some studies, including obesity, sensitivity to asthma. We need more information to be more affirmative on this.
Yeah, just to respond to this, I think the problem is that we do not really know the mechanisms behind this programming effect so far. So we have some ideas, and we have a lot of associations, but we do not have a mechanism. With respect to the diabetes increase and the microbiome, this seems to be very interesting. There are studies in rats at least, where they took from an adipose or a fat rat the microbes and they injected them into a germ-free pup, those microbes, and then they found out that those perhaps develop adiposity by themselves. So there seems to be a link and an association between the microbiome and the changes of the maternal diet and the changes in the microbiome.
Yes, sure.
Those are studies from the McMaster University in Hamilton, just came recently out.
You're absolutely right. Then the microbiome itself acts on the recipient through epigenetics marks, and so it may well, it's an important factor, but it may well be a vector of these changes. It depends on at which level we consider the complexity of this causation at the level of a system like the microbiome or at the molecular level. And it's certainly an important actor in all what we mentioned here.
Maybe just to share a philosophical thought on that whole topic. Of course, it's very interesting for health insurances to have a lot of normal people. And it's, I think, very important as a physician or any human to know that everything, every decision, every intervention, everything has an impact. Often we don't know what impact it has. And but we will never have 100% normals because we will always change that. And it's, I think you had this nice diagram with the physical, mental, and social disorders. And I think we all need exactly those individuals because evolution wants these individuals because they make a difference. I mean, if you look at people like Goethe, Schiller, Beethoven, Mozart, Humboldt, they had all of this. They don't breathe well, usually, and they die young. Not all of them were very old, but a lot of them die very young. But they make a big difference for society, so they have just a different job. Yeah. You know? So I think also it's interesting to look in that way on the whole topic.
Sure. I fully agree because epigenetics and programming are considered today as possible also factors in the evolution, the general evolution, which do not exclude the Darwin approach, but and also I would say that there are, well, this shows also there is the definition of normality is questionable. Because we have such a continuum between, even within the normal range, and of course, changes which, the whole story is about trade-offs. You gain an advantage and you have to trade to accept a disadvantage on another side. That may also be one point why the very famous people you cited are so excellent.
Well, not only the definitions of normality are wobbly, but also the definitions of pathologies. So we are all aware of diabetes 1 and diabetes 2, and for the past 15 years, diabetologists have been starting to talk about diabetes 1.5, which is in between, which is particularly regards early onset diabetes, which is not typically diabetes 1 nor typical diabetes 2. So could that be maybe what you were just presenting, be a source of this diabetes 1.5 that we see growing and increasing?
I think as doctors we need to define models for reasoning, to make decisions. So we need, but we need as well to keep in mind that each person is an individual, and even the 95% confidence interval, it means simply that 5% of the individuals which are not in the interval are normal. So it's a relative definition. I guess we need these models to define the disease. The nature did not know medicine, so we need also to be able to think out of the box, even in front of the patient, and define also for mental and social disorders when there is suffering. When a patient is suffering, then this could be a good reason to act as a doctor instead of a quantitative threshold or something.